Specialization that we don't buy, so just give it up. All right, the wildebeest group will... Their project. Where are the slides?
Well, we're going to start with...
Who stole the microphone?
The video
Okay, we're actually going to start with a little video, which we can't embed into the PowerPoint, so instead of coming in and out.
That's for your autobiography, that you write to Jim. Show it video.
Here we go. You might remember this from last week. And we still have no sound.
Oh, we need sound.
Imagine some squeaky tamarin noise. So these were the tamarins that were cooperating with each other, even though they didn't quite have to, by sliding the tray in and out. And it was kind of the inspiration for our talk. Oh, our project, which we're now going to talk about. So, opening slide should be nice and clear, because the beginning of the talk is some theoretical background. So we were looking at co-sociality.
So you have a hierarchical structure in your group.
It goes reversed alphabetized and then alphabetized in order to counterbalance the alphabetization. but ensure that your PhD student comes first. There we go. So co-sociality and human-robot interaction. How does an individual's level of agreeableness affect perception of a humanoid robot giving positive or negative feedback during gameplay? Okay, so this is going to take a while to break down. So what we did was when all half of BCBT got together in our group, we started to think about what had inspired us over the previous time that we've been here in BCBT. We were looking at Asif's work in vocal cooperation and turn-taking, which of course Diana works alongside, and the pro-sociality components, which was the tamarin video that we've just seen. My own research is very much focused on the importance of individual differences in human-robot interactive research, applying psychological measures in an appropriate way to a new discipline which involves this non-human artificial social agent. And of course, we had the fantastic resources here at Specs, people with a background in empathy and with some pre-made game-playing, turn-taking games with the iCub and Vicky's vast knowledge. So combining all of these together, we decided that we wanted to explore personality in HRI and demonstrate how the individual personality of a human is a key factor within human-robot interaction. So, of course, we sat down and preliminarily decided, what do we have to work with, okay? So we had a lot of experienced coders, C++, PHP, etc., and we had some great statisticians, experimentalists, psychologists, which was great, an excellent web developer there in figure 1, and as I said before, the iCub and the Reactable, okay? And the whole thing started to develop at this idea that the iCub had this turn-taking game, which we could manipulate as part of the variables that we were going to start to look into. So we had our bunch of geniuses and an iCub and React table, and we decided we wanted to create an experimental design that was going to allow us to explore the effects of co-sociality, which is how we'd interpreted pro-sociality, which I shall get to in subsequent slides, understood again as this individual difference component, which we could explore via a manipulated interactive period with a social agent that's artificial, the iCub. Now, part of understanding and developing good HRI methodology is the systematic testing of psychological theory. So in whatever way you can, to go through and look at these metrics that are normally used in the literature, over the last 100 years in human-human methodologies, and exploring them again in human robots. And with that in mind, that's great, because then any kind of result is an interesting result, because if it's going the same way, then you can say, well, it looks like this social agent is being treated by our human as another human, and if not, we can start asking, well, what is a robot? Okay, so I'll go back to the foundational aspects. So we decided to create one of our divisive variables by looking, again, at individuals, which self-select, which is fantastic. So co-sociality is also called cooperation, and cooperation in psychological literature is measured via agreeableness. Now, agreeableness is one of these 5 broad dimensions of personality, which is otherwise described as OCEAN: openness, conscientiousness, extroversion, agreeableness, and neuroticism. This big 5 inventory has been systematically and cross-culturally validated over the past 40 years. It's a very, very solid psychological theory, which is brilliant, you know. So you can start your experimental design with something you know is pretty much on the ball there. So we extracted from that this fantastic 10-item validation questionnaire, and from that we're going to start to divide up our group of individuals based on how pro-social they are. Now, agreeableness can be seen as a continuum. So individual differences along this continuum start looking at social harmony in either a compassionate and cooperative way, and then they move all the way along to people who are kind of suspicious and antagonistic towards others. Now, again, within the literature, this has been explored quite extensively in team working scenarios in order to see how humans get along with one another when they're receiving positive and negative feedbacks from the world around them. So we thought, that's great. We've got this turn-taking thing with the iCub. We can easily write some kind of new configurations to get our iCub to be in the positive cub or the negative cub configuration. So now we have two things to play around with. We have our self-selecting participants who are on this agreeable continuum. We have an iCub with which our human participants can now play an interactive game with them and this artificial social agent, turn-taking. And then we have our iCub in one of two conditions in which it is treating our participant either terribly nicely or not so nice. Now, some examples of the sorts of phrases that we started to build into the iCub's response is put up here. So we have, you know, participant answering correctly or incorrectly, and we have our positive iCub sort of saying, good job, or hey, we're having fun, don't worry. And then the negative iCub example, which, trust me, we went through a lot of iterations for to make sure it wasn't too rude, but rude enough. Fortunately, I'm British, that comes naturally. So we had, you know, participant answers correctly. Do you have any friends? Do you just stare at maps? It's really hard to get sarcastic intonation in a robot, but we tried. Participant answers incorrectly, simples. How did you graduate from high school? You know, etc., etc. And this, again, is grounded in some literature in which our agreeableness ocean big inventory has been cross-culturally explored with these positive and feedback team-working scenarios. So this is really nice. Now, it's important to note here that in human research, the consensus is that in order to care about another person's opinion, you need to value that person's opinion. A robot, contentiously, I know, in a room of, you know, roboticists, but contentiously, I'm sorry to say, a robot does not have an appropriate level of social agency for a human being to seriously value that social agent as a mind, as it would akin to a human partner. Or does it, right? So we had this really nice framework to start at in which we could now start to build our experimental ideas in which we address psychological theory in HRI. Now, in order to explore agreeableness and the positive and negative feedback, we need a way of measuring this. So the final part of building our experimental design was thinking back to these tamarins and their task of turn-taking, and also the other little tiny primates, Diana? Marmosets, that's right. The marmosets that were willing to sort of pull this lever out. And was it Asif talking about that last week? Yes, Asif was sort of saying these little caged-up marmosets are opening and closing us. So we kind of wondered that if agreeableness is linked with compliance, which it is, again, extensively in this wonderful pre-ready-made psychological literature, if it's linked with compliance, how can we link this idea of people being more or less agreeable to a post-interactive gameplay scenario with your iCub to do a task that we could consider really boring, which we could measure on various variables, whether or not that scenario with the iCub was having any kind of effect. Now, again, we were well aware of the constraints of the time. In order to get any kind of power in your results, a single variable needs at least 15 humans in order to work. So we're talking now with 3 sets of 2 variables. This is numbers that are beyond my capability, but it's more than 6, right, on a bank holiday in the middle of a quad. But we did manage to do some quite exciting stuff based on our experimental question. A slide with loads of text, always a good move. So after playing a game with the iCub, providing either positive or negative feedback, do humans who score more highly on agreeableness rate expressive facial images of the iCub, which Jimmy's going to get to in a moment, with higher valence, which Jimmy's going to get to in a moment? And then conversely, are those that are scoring lower on agreeableness more affected by negative feedback? So if we go back to the psychology literature, we have people who are kind of rate high in agreeableness are perceived by others as more able to just kind of ignore the antisocial people around them and are much happier to get on with any kind of task. And so we're kind of saying, you know, if it's a boring task, are these more agreeable people even happier to just spend time on it, to spend more time looking at what a valence would be? Where you're less agreeable people, maybe going to get irritated by negative feedback, are going to try and finish that boring task quickly, and not going to be as precise as they could otherwise be with their measurements. And this would be the sorts of things that we could then explore. Again, in quite a nice broad idea, because we're trying to translate a psychological human idea onto a robotic human idea. So again, our initial experimental hypothesis is quite broad and underlined by this. Should we even be doing this? Should we be treating humans and robots like humans and humans? Okay. Experimental methodology, my second wonderfully long slide. Guess what time in the morning I did this? Imagine there are pictures. So we wanted to make our methodology really tight, okay? So we obviously started with our consent form. Ethics is very important when you're playing around with people. Then we had our participants entering the iCub room, in which the iCub alone gave them instructions in one of two configurations. At the very end, the iCub asked our individual to leave. Now, the final phrase that the iCub said was the same in either condition, which had no negative or positive connotations, in order to avoid any kind of recency effect. So often, if you're with someone and you're having a bad time, if the last thing they say to you is really nice or really horrible, conversely, that's what you're going to go away thinking. So we kind of tried to, what's the phrase? We tried to control for that in the little ways that we could. We also, we also had to make some baseline data on the iCub rating, because obviously if that's going to be our measure, we can't just throw people in and say, well, we're going to compare two groups. So we did some baseline data in order to understand what people who hadn't met the iCub were thinking about these facial ratings. Jimmy.
So as was just mentioned, for our baseline data, we wanted to make sure that just interacting with the iCub isn't going to dramatically affect how much time they're spending on this task and the way that they're rating these faces. Now, these are iCub faces. We actually took photographs of the iCub, we used, of all the different possibilities that we had, a subset. So the eyebrow, which you can see the eyebrow, actually it's the last one to move, but it has 5 different configurations on each side, to where there's actually 15 different configurations you could use. The eyelid has 5 different levels of openness, and the mouth has 6 different possibilities, which is scrolling through here. Now, to those of you who actually did kindly enough volunteer to be participants for our study, you could imagine how if we had 150 combinations to test, that would have succeeded in being a tedious task, but maybe would have been a little bit too much so. So we tested 36 faces, and they were the ones that were either the most neutral or with the biggest smile or the biggest frown. The biggest, I guess, variability in eyebrow configuration, along with the, eye completely closed, completely open, or intermediate. So we had the largest range of faces, and all the ones that we used are right here. So those of you who are volunteers might be currently getting PTSD. Now, the way we did this is we tested these people, and they had these 4 different bars that they could move, 2 of which measured discrete variables and 2 of which measured continuous variables. So we have valence, positivity, excitement, and arousal. Valence and positivity are measuring something similar, but in order to make sure that we have the external validity and we can compare our data to the literature of emotional state, we wanted to make sure we included a discrete and a continuous variable for that, along with excitement and arousal, going from calm to excited, or the robot with just one little dot to the one that's nearly exploding. Again, for the same reason, we want to make sure that in all the spectrum of emotional states that this robot is trying to display, we can compare that to the literature that's out there. And we did this exact same task for our pilot data with people who never interacted with the iCub, along with to all of our participants who are in either one of the 2 conditions which we discussed.
Oh, cool. And the other thing that we were controlling for was empathy. So obviously, again, to go back to the looking at humans and humans together, or humans and robots, one of the reasons that human-human interaction works so cohesively is because we have this level of empathy. Of course, if a human being is going into a social scenario with a robot and doesn't feel any empathy for that robot, nothing else in this scenario makes any sense, because we're now showing that the psychology cannot translate. So Maria, who is a master student here, has an augmented version of the basic empathy scale, which is, there's a couple of example questions here, which are all about the robots. So we gave this to our participants after their interactions to then be able to compare all of our final results to whether or not they were actually empathizing with the robot as a social agent, and therefore whether or not we could take anything else for, with anything more than a pinch of salt. Skipping, video of interaction. So this is what happened.
Hello, I am VQ. Let us play a game of geography. You and me will play in turns. Each turn one has to select a capital and match it with its country. My turn. Moscow is the capital of Russia. Let us see how you do.
Ha! Athens is the capital of Greece.
You did not seem confident with that, did you?
I was.
Nairobi is the capital of Kenya. Choose. I want to laugh.
Huh. Accra is the capital of Ghana.
Do you have any friends, or do you only stare at maps?
Asmara is the capital of Eritrea.
Are you cheating? Because I do not understand how you got that right.
I guessed.
Moscow is the capital of Russia.
I can learn from you. Now it's my turn to play. Reykjavik is the capital of Iceland. Your turn.
Seoul, South Korea.
Wow, that was impressive.
Nairobi,
Well done.
Maseru, Eritrea.
That was difficult. It is okay.
Thank you. Maseru is the one from Ghana. Wrong.
I am sure you know lots of other stuff. It is okay.
And then participants proceed the questionnaire based on this final sentence. Okay. Oh, crumbs. Okay, so now we can kind of get to our results. And I'm going to pass over to Diana. Oh, yeah, sure. It would probably be due to time constraints, but you would have to ask the mighty Vicky.
There wasn't. It was just in that specific scenario where we were making the videos. It wasn't run. It got stuck. But normally it wasn't fixed. Yeah. It was everything, but just this only in this specific video because we had to do one quick.
Diana.
Okay.
Yeah.
So now we have results. We mentioned before that we did a baseline of people that never interacted with the iCub, and also because you guys may not be indicative of the normal population. So on the x-axis are the four ratings that we had: arousal, valence, excitement, and positivity. And on the y-axis is the 1 to 7 that they're rated. And you can see that most people rated collapse across all the faces. Most people were around this area. So now for the exciting part. This is the people that interacted with the nice iCub and the mean iCub. And from other figures on, I'll be using the same color scheme where blue is nice and red is mean. And the darker dots here are the points for individual subjects, so n of 6 in each. And then the lighter dots are the sample means. And there are no differences across groups of their ratings. Okay, But what we were really interested in is this agreeableness measure. So here is the group data, the people in the nice condition, in the mean condition, and their agreeableness. And these are the distributions of people's agreeableness rating from that Big Five questionnaire. Okay, and so what's interesting now is the data, the correlations over here, where the x-axis is agreeableness, and the y-axis is their rating. So we can see that on rating the ICUB faces for arousal and excitement, there's no correlation. It is pretty much flat. But what's interesting here is on the ratings of valence and positivity, we can see some sort of interesting trends. For example, the people who were in the mean ICUB condition and were less agreeable tended to rate the ICUB's faces as less valent or less positive than the people who were more agreeable. And conversely, the people in the nice ICUB edition that were less agreeable tended to rate the faces of the ICUB as more valent or more positive than the people that were more agreeable. And you can see that across both valence and positivity. We ran preliminary stats on this, but given our small sample size, it was not significant for both the correlation itself and then the difference between the two correlations. Okay, so we mentioned that we had 36 different faces, and then we can apply, the previous analysis was collapsing across all faces, but we could also apply the same analysis looking at specific features. And so one example of such an analysis would be looking at the eyebrow configurations, where these are slanted down, these are flat, and these are slightly raised towards. And to do that, we can look across groups and then show these box plots. In the interest of time, I won't go through any of them, but you can look at them. And also, we can then do the correlation of agreeableness and all these ratings across features as well. And then we can see some interesting correlations here.
Anything significant?
Nope, p equals 0.5 for most of them. It's okay, the group data I showed was p equals 0.9.
It's okay.
Yeah. But not surprising given our small sample size. So I've talked about the different ratings that people did, but another measure we also looked at was the time spent rating faces of the ICUB, where before we were like, you know, how long are people willing to do this or take the time to do this carefully for us? And here is the group data. We have the nice ICUB and the mean ICUB, and then the time spent. And then in the group, there was no significant difference, but then we do this correlation with agreeableness, and we see this trend where people who are less agreeable tend to spend less time rating these faces than the people who are more agreeable. And this is different by the condition. And finally, we actually do have a significant result. This is on the robot empathy questionnaire. Not surprisingly, people who interacted with the nice robot tended to be more empathetic than the people who interacted with the mean ICUB. And we, again, do the same correlational data here, where if you were less agreeable, you tended to be less empathetic.
Wait, you mean
Okay.
people scored higher on the question whether the robot is empathetic, or?
Okay, so people scored higher on this empathy questionnaire when they interacted with the nice ICUB versus the mean ICUB.
About the robot being empathic or not, right?
No, yeah, we didn't.
About your feelings of empathy toward the robot.
Yeah. Okay. And then, so that's the group data. And then the correlational data means if you're less agreeable, you're less likely to be empathetic towards the robot. And I get to pass this on.
Oh, cool, beans. Okay, right, I'm on 22.9. Okay, results in sum then. So as Diana is saying, obviously we got far too low a power for any kind of statistical anything to come out of this, and I'm not going to make any judgments on this, but as a pilot data, it's definitely intriguing enough that the interesting results that we see caveated against this low power would make it something we would want to explore further, and certainly opens up this question again that using these human-human inventories and human-robot interaction doesn't seem such a silly idea. So again, we have these, you know, for example here, in both groups, those who scored lower on agreeableness score lower on empathy, which follows the literature, because these are two scores that are correlated against each other. More agreeable people with a negative ICUB spend more time rating their faces and rating them with higher valences, which sort of goes in the direction of a hypothesis, but I'm not making any statements because there is not enough data for me to do that. And then a project final thought, which I think is the most important thing to take away from what we've done here in just a few days, is that this Big Five inventory and basic empathy scale literature, which has this massive established history, and which allowed us to create this really nice initial theory, in our personal opinion, allowed us to ask this question in which we could see whether, tackling human-robot interaction in a human-human interaction kind of manner, where we treat this robotic social agent as a social agent worth discussing in human terms, seems to indicate from these early pilot results that we can be doing that. And that made us all terribly happy. And I would like finally to say thank you to Maria, who we couldn't acknowledge on the first slide for technical reasons. But any questions? That's it. There is a pre-approved list of questions, but we can just exit that. That's fine.
So now, look, in some sense you could say some of this is sort of circular, right?
Mm-hmm.
For instance, if someone would be more or less agreeable, they might, let's say, have a broader range or a smaller range of the acceptance of the behavior of others, and as a result, they might be more or less empathic, etc. Right? So how do you control for that circularity?
I'm actually going to say it quite naturally. That's a really interesting question, and I mean it.
Why do you have a group of 8 people and you have so few people presenting?
Because we were asked that only 2 of us could present.
Oh, really? Okay.
Yeah, it was Anna kind of, yeah.
I didn't know there was a limit to the number of people who could present. I thought everybody could present.
Yeah, yesterday we were told, yeah, limited to.
Organize it, but it has to be concretely within time of the. Okay.
All right, doesn't matter. The question is still, how do you control against circularity? So then just anyone, whoever has the answer.
I don't, but I want the answer.
Good, then you're ruled out. Does anyone else have an answer? No, but how will you in the future? How can you in the future?
By extending the paradigm, I imagine, in some way. Damn it.
Okay.
Oh, no, I'm going to be thinking about this for hours.
Another point is, the properties of the robot that you controlled were the facial expression and the speech. But now in the speech, so you have the content of the speech. And there, in some sense, you have just sort of invented utterances, right?
Well, they're kind of a little bit based on the a paper that we did for Living Machines for EASL, in which our Xeno R50 robot, which we were running some interactive gameplay experiments with in a museum in Sheffield recently with a bunch of children, also had this positive and negative, obviously much nicer because they were children, responses built into them. So we kind of built onto that.
No, wait, exactly.
Yeah.
The point is, how do you now validate that these utterances are really, let's say, negative or positive? Like of course, you can use your own intuition.
Systematic testing, I would say, over a very broad population. Yeah, go on.
No, we haven't really tested about that, though. We should have a pre-test of people randomly evaluating its sentence, so that we would be able to choose within, like, let's say we have a.
So would you have these inventories, right, of emotional connotation of single words? Also, Daniel has been working on that. Right?
Yeah. No, it's true that we haven't used it. Actually, to be honest, we had so much fun picking our own images.
Really, really hard work.
Phrases. Yeah. If we do it a bit more seriously, we should definitely.
Okay. Other questions? Okay, then thank you very much.
Too much fun. That's mine. That's mine. Yeah. Oh.
Who's up next?
change this one, right?
I sent it.
No, I didn't.
I sent it.
I'm talking about This is from like an hour and a half ago. We're missing some people. Yeah, maybe we can present from that one, right? Why not? You've got this.
No. Which? I got it actually. This one.
Sorry, typical technical
C'est le
problem.
Venom BCBT', this one.
Yeah, but that's an hour ago.
I got that photo. You wanna make?
I thought you changed. Did you change the slide around?
No, it's okay. Which one is it? Open it.
I thought. This slide. I thought this was being moved before the program. Yeah, exactly. It was. Has it been moved?
It was
But it doesn't matter. Let's present it. Okay, I don't know how to tell this story.
No. ¿Cómo que no? I don't know, I sent it. You sent it? Yeah.
So while we stop, a brief introduction to the project. This is a follow-up of a work that we started like some time ago on investigating the capability of the cerebellar microcircuit in acquiring anticipatory adjustments to posterior perturbation in a setup that reproduces the dynamics of the inverted pendulum. Does anyone know how to full screen it?
Just maximize it.
Good view. This is.
The last ones on the bottom.
Oh yeah. All right. Yeah. So, what I was saying, this is a follow-up of a project that we already started, a stuff that we started already investigating on the capabilities of the cerebellum to acquire anticipatory responses in postural tasks. So we investigated in theoretical terms the setup of the inverted pendulum and checking how the cerebellum can actually acquire anticipatory responses that reduce external perturbations, predictable external perturbations. And now the follow-up to this previous work was, let's say, double. So on the practical terms, we wanted to implement this control architecture based on cerebellum on this self-balancing robot. So since we were quite a big group, we split into, thanks Santi, we split into into subgroups, and some people worked on the robot, so made some improvements on the hardware and on the basic processing that actually happened on board. On the other hand, some other people focused on improving the cerebellar algorithm and to actually get it ready for the real world. And in order to do this, we decided to focus on adaptive strategies for the learning rate modulation. So basically, usually this algorithm uses a constant learning rate, while we thought that it was worthy to give a try and also try to benchmark like different strategies for adaptive learning rate. And in this case, with some other people, we focused on comparing different strategies, which are like a fixed learning rate, an exponential decay learning rate, and then some other two algorithms that are dynamic algorithms. One is it's a classic algorithm for this scope that is called ball driver, and the other one is an algorithm that actually we took inspiration from a paper from Paul from '91 that is called chaos-based learning. So we gave a try to that one as well, and now we're going to have a brief introduction, an explanation on the robot and what worked and what didn't. And then we're going to have like some theoretical results on the comparison of the different strategies. So, Carlos?
No.
Oh, Deepa?
So how do you move two slides on this one? How do you go to the next slide?
Right. Thanks. You should do it every slide. No? I was like, okay.
Okay, so Giovanni mentioned all of this just now, but just to state our goals clearly up front, we wanted to implement a model of the cerebellum that uses different learning strategies to counterbalance perturbations in the environment. And then we wanted to test whether a dynamic learning rule leads to better performance than a fixed learning rule. So briefly, to give you an overview of the biological circuit of the cerebellum that we're interested in, we have ascending input from the mossy fibers here, and that synapse onto the granule cells, and the axons of the granule cells form parallel fibers, which then synapse onto Purkinje cells. Purkinje cells also receive input from the climbing fibers, which originate in the inferior olive. And this microcircuit is believed to be responsible for learning during classical conditioning. So an example of a classical conditioning paradigm is eye-blink conditioning. And basically, what you have in this paradigm is you play an auditory tone, and that's followed by a puff of air to the eyelid of an animal. The auditory tone would be your conditioning stimulus, while the puff of air is your unconditioned stimulus, and the animal responds to the puff of air with an eye blink. After many trials of training, just your conditioning stimulus, which is the auditory tone, is sufficient to trigger the eye blink in the animal. And the response is triggered in anticipation of the air puff. So a major site where this kind of learning is thought to occur is in the synapse between the parallel fibers and the Purkinje cells. The climbing fibers encode an error signal, which carries information about the unconditioned stimulus. And when you have correlated firing of the parallel fibers and the climbing fibers, you get this modulation of the modulation of the strength of the synapse between the parallel fibers and the Purkinje cells. And this plasticity is what is responsible for learning that takes place during classical conditioning. So now that you have an introduction to the biological circuit, Carlos will tell you about the model of the cerebral circuit.
I don't know how to do it. Here. Okay. So we are now going to make like this bridge from the biology, like the structure of this microcircuit cerebellum, to the actual model of the cerebellum that we have. And this is based on a paper from Ivan. Hey, here. But also from the Puril one, no? Puril?
Yeah, but this version is from Ivan.
Okay, so thank you.
It's better actually than that one.
We took the best one. So, okay. So this cerebellum model is like understand like as if it was like an adaptive filter. So we have some processing of the sensory information from the input from the granular cell layer that is interpreted in a bank of filters over there. And then we have like the Purkinje cells output at the end that we model it as a weighted sum of the parallel fibers. So yeah, the weights correspond to the synaptic efficiencies. And we also have these climbing fibers that correspond directly to the teaching signal that adapts actually the synaptic weights using the covariance learning rule. So formally we can see like the formula there where the change of the weights is equal like the learning rate multiplied by the teaching signal and the signal that goes to the weights. And that thing, so the main thing of our project is, okay, yeah, try to make this beta fixed parameter in this model dynamic. So we have different strategies, and we're gonna talk about this.
What is the aperture?
No, G is the granular cell layer.
But isn't it then why is scale into one situation goes to many and then back? So it's like maybe the question is for us. No. What is the question?
Why do you have so many and then back again to one?
Yeah, it's like parallel situation happening that again go to one.
Yeah.
Because this is like the expansion of the input into different temporal profiles. Okay. So basically you decompose the signal. This is like the analysis part of the algorithm. Okay.
But it's not related to prediction, no? It's not related to prediction.
Well, it's related to prediction, but what you actually learn to predict is the signal that is coming from the teaching signal, so the one that updates the weight. But basically, the expansion of the input in the analysis phase gives you a set of components that then are going to be weighted and summed up to then give like the predicted signal. So basically, these are like the building blocks of your signal at the level of the algorithm. But this actually maps pretty well with the physiology, because like you see like a similar structure, a very similar structure, and in the granular layer, in the expansion with the parallel fibers, and then the synthesis part would happen in the level of the printed cell.
Okay.
Could you explain a bit what is the composition of the signal?
Basically, it's safe. You have a certain stimuli, and that stimuli, the composition is so that in the dendritic tree of the Purkinje cell, it makes many synaptic contacts. But the cell is one, so this output is like a synthesis of all the different contacts from the arriving parallel fiber.
Imagine equalizing the sound, so you get the sound and you compose the frequencies, you do some sequences and then you mix it again and you get the sound again.
Yeah. And the important thing is like that is happening here is actually how time is managed. So like each one of these let's say component has like a different temporal profile. Okay, and this allows you like to move in a range instead of timing, so that suits better the reproduction of everything.
Okay, Giovanni, you know what you're talking about, so can maybe give an explanation?
Okay. So this is the setup that I was mentioning before. So this is just a schematics. In order to investigate like the role of the cerebellum in this case, we're going to use this inverted pendulum setup. We have, as I was saying, we work both on the robot, both in a simulation. So this is just a schematics. It will be like the reproduction of the self-balancing robot that we have there. And what we have here, basically, we have like a cart that is moving on 1 degree of freedom, just basically left and right. And the goal is to balance a pole that is mounted on top of the cart. Then a perturbation is thrown to the pole, and the cart has basically to counterbalance the perturbation. So this basically can be achieved like in a reactive manner. So using a feedback controller, that is the bottom part of the diagram that you see on the right. That's a PID controller that will take the actual angle of the pole and a target angle that in this case is like the vertical stable position. We'll track the difference of the two, and basically we'll try to correct the error once that the error is perceived using, well, the PID logic that I'm not gonna throw now, but basically it's a way like to decompose the error in different components. And the idea is that the reactive controller is gonna output a motor command that in a compensatory manner is going to adjust the pendulum position. But then we have the cerebellar component on top of the architecture that would act as an inverse model in this case, in a sense that it's going to be able to associate a given action outputted by the PID controller with a predictive signal that is a proximity sensor signal that basically is signaling an incoming perturbation. And the idea that taking also the kind of learning that we see in classical IB conditioning, the cerebellum is going to output an anticipatory response that is going to anticipate the response of the feedback controller, and basically is going to compensate for the perturbation in advance. This is learned in trial-by-trial manner, so the cerebellum doesn't know anything at the beginning, and it actually learns from the arrow signal, that is that arrow that crosses the cerebellar box. And basically, what the cerebellum does is finding like a correlation between the proximity signal and the arrow signal coming from the PID. And so we have like a part on the robot, and then we can look at a adaptive learning rate algorithm that we implement.
All right, so I'm gonna talk a little bit about the robot. Obviously, we didn't have like a, yeah, this is the robot actually, that you can see conveniently depicted in this picture, but it's also here. So the robot basically has like a principal axis with 2 wheels and 2 motors that can use to, apply some motor force to it, and it has everything I'm gonna say is like pretty much there, but I'm gonna explain it here because it's like closer. And on top there's accelerometer and a gyroscope that we will use to measure the actual angle it's inclined. There's some electronics. This is handled through an Arduino Mega board, and there's also the proximity sensor that we can actually use to measure. It's an infrared sensor actually that we can use to measure how close the perturbating object is, so that the cerebellum adaptive layer can actually anticipate and act in consequence. So what are the parts that we did for these robot control thing? We implemented a simple yet effective PID controller, and to filter the noise, that the inherent noise coming from the accelerometer and gyroscope signals. We implemented a Butterfield second order low pass filter, illustrated in the figure to the right, with fine-tuned parameters such that it gave a nice filtering but not a lot of delay and a good resolution. And then to integrate the measurements by both the accelerometer and the gyroscope, we implemented a simple complementary filter that allows us to calculate a more reliable angle estimation. But hardware is hard, so we had some issues. First, it's quite difficult to calibrate the 2 sensors at the same time because it's a noisy signal. There's drifts, there's noise, there's quite a lot of things to take into account. So it didn't quite cut it. And then there's also a coarse motor response. That is, the smallest motor command we could send to the motors was already too large to get a fine balance of the robot. So our capabilities of robot stability was a bit reduced. So basically the main results we have for this project are made in a simulation. So at this point I'll turn to my software colleagues to give the details about it. Is there a point? It is. Which one? The main one?
Green one in the middle.
It's not green. Cool. I don't know this. Okay, yeah, so this is the idea that we were going to adapt our learning rate so that if our model is doing well, we adapt it to try and slow down the learning or improve the learning depending on our strategy, and compare 2 different adaptive learning rates compared to like the really basic fixed learning rate or decaying learning rate. So this is design. So this is how we expect them to go like in terms of how well. So S1 being fixed learning rate, we've seen that was like baseline. Exponential decay, we would expect to do better. And then bull driver, the strategy here is if you're doing well, you increase the learning rate, so you take bigger steps towards the solution, where you hope the solution is. And chaos-based is you kind of ramp up the learning rate to begin with until you find yourself in a chaotic state, and then you kind of try and slow down the learning rate to in a certain strategy that I don't really know, that helps you try and find the best local minimum, so the global minimum. And then the main test we did was just with fixed perturbation, so we didn't change between trials. But then afterwards we looked at maybe increasing the strength of the perturbation in blocks. So every 30 trials we increase the amplitude. Here we have learning curves. So the area we're measuring was the angle squared summed across all trials summed across all time steps per trial, and we plot this against trial. And as you can see, so adaptive beta here is actually what we call the chaos inside the code. So this did the best, and then the board driver, and then the fixed and exponential decay, which, as we hoped, weren't as good. To the right, you can see how the learning rates evolved over trials, and red is the board as the chaos, which did the best. So you can see it ramps up the learning rate and then comes down. The board had a strange, opposite strategy, which was to just increase the learning rate. Not really sure why. So this is comparing. So this is the first trial and the last trial for. This is the VIX beta. So the green line is the motor drives from the PID, which is our reactive component, and the blue is the cerebellum, which is the adaptive. So as you can see at the start, there's no adaptation, it's just working on the PID. And then at the end of the trials, there's some compromise where they're working together, and you can see that there's this anticipation period. So the dotted line is the onset of so when it first sees proximity, the dashed is the time that it hits the robot, simulated robot. So these are the final solutions for each strategy. You can see they all kind of converge on the same strategy. It's the rate at which they converge there. The two chaos and bold actually do something strange where before turning to the left, they turn to the right. I don't know what strategy this is, but maybe it's setting the robot up at a right angle. So this is just how we looked at the blocked condition. We didn't do too much analysis on this because we didn't have the time. But you can see that the different strategies behave differently. So it's clearest in the fixed. So each phase trials, you increase how strong it is, so the error increases, so it has to relearn. And we found that the board drive was actually the best at learning this. Although if you look at the slope here, then maybe the adaptive is better at changing, it's just not got to the solution early enough. Conclusions. Changing the learning rate in a dynamic way does actually help, which, yay, good. In particular, when looking at the fixed perturbation, the board drive and the chaos were a lot better than the two fixed and decaying. I think we need to look more at the kind of like the fitness landscape of this, because we didn't really understand if we were in a convex or it looked to be mostly convex but quite bumpy. So when learning rate was small, it was getting stuck a lot. I think that's it. Cool.
So,
how did you actually create the chaotic dynamics?
Sorry.
How did you create the chaos in your model?
Following basically your algorithm, we were like checking out the variance of the error. We were like trying to increase the variance in the error. As soon as we get actually we didn't get to a real chaotic state. Like our criteria for.
Yeah. No, what I was saying is that the phases that you have, well, as you probably know since you wrote that paper, in these algorithms, to rise a lot the learning rate until you are able to assess a chaotic state, and then to cool down basically in order to avoid, let's say, local minima. So in this sense, we followed your algorithm, and the idea is to increase the variance in the.
Yeah, but wait. The point is, in your case, you are also a controller for this robot. So if you increase your learning rate, it also means you're changing your actions, or you're not changing your actions.
No, of course we're changing.
Exactly, and I think this is the big difference. Like in the original paper, what you exploit are just you create positive feedback between the units of your map, and that's the source of your chaos.
Okay.
But what's cool in your solution, the chaos comes really from the motor control. This thing starts to wiggle, I guess, in some weird pattern.
Exactly.
So you start to create chaos. You have a chaotic exploration behavior, if you want, of the state space, which is really cool. It's very different from the original paper, where it's all internally generated from. Okay.
No,
so it really comes from the action. This
no,
is
it definitely comes from the action, and in that sense, we assess basically there is a threshold, the variance of the responses we can get, and basically this would set like the upper boundary for the learning rate, and then you start like the cool down phase, in which like then you actually enter hopefully in the real local in the
Maybe
real.
so like you said in the conclusion, the error landscape. Do you have any feeling for that? How does it relate to your actions?
Well, this is something that we should actually investigate. So it requires like a more, I think, analytical study, because like there are definitely, I think, local minima that this is like given by the configuration of the bases, no? That we instantiate. So I think that the error landscape really depends on the number of bases that we use, because this is basically the substrate, you know, that we're going to use to then recreate the error signal. There is also another thing that it would be interesting to investigate. That is the interaction with another parameter of the cerebellar model, that is the nucleolivare inhibition. And this would go like in the direction of like the work of Ivan, no, in this idea of and Santi, in this idea of like this cost function, no, that is also updating another parameter. No? So an interesting thing would be like to see if there's like a competition between the two strategies, or some sort of like syntony. And this is something that would be nice to see.
No, it's really nice. But then the other thing, anyone is aware of, let's say, in any kind of animal, a codic exploration of a movement pattern? I have an example. I will not tell you yet.
Okay. Well, I didn't. Yeah. I mean.
No, they're too young. They don't do it.
Okay, but don't you think that the motor bubbling? I mean, it's not really chaotic, but still,
well, maybe
it's an exploration of the state space, no, in the motor domain.
Right.
So yeah.
It might be interesting. Maybe we could have a human learn the same task and to see what's their exploration strategy.
Right.
Whether it has, let's say, hypervariable phases.
Yeah, exactly. No, it's like, cool, yeah. I mean, this is definitely like I think the exploration of the motor state space, no, to then really cool down to the optimal solution. No, but you have like to know the state space first.
Yeah.
No, so this is like I think the interesting component of this.
Right. Other questions?
This is great.
Ivan asked a question.
Yes. If you have maybe you have browsed the literature on adaptive learning rates, but in the field of neuroscience, what would be the neural substrate? Yeah, substrate for this. So
actually, we have an answer for this.
Yes.
Right. So.
So, I mean, in the hippocampus, they've shown that you can modulate the rate of synaptic plasticity based on previous experience of stimulation of LTP and LTE. Both has been demonstrated in the hippocampus. I have not found any literature in the cerebellum.
But there might be then.
But the easy thing to point is always neuromodulation.
Right.
Neuromodulation is a good point to start.
Yeah.
Right, like acetylcholine in cortex is really changing signal-to-noise ratios and so on. So, and also you have a dopaminergic innervation of cerebellum, so that might be a good place to look. That's because you know it has an impact on plasticity.
Yeah.
Right? It's very good.
Okay.
Wonderful. Other questions in the back? In the back. No, they're all sleeping. All right, thank you.
Thanks a lot.
What is this?
All right. Great job, Yvonne. Very good. Very good. Thanks, Jose. Thank you very much. And then, you better.
Oh no. Oh. I don't have this. And I don't have this. I need to take it. But I don't have this. I need a DBE.
You don't have it?
I need HDMI. HDMI.
Oh. I don't know about this.
Look, this. Do you check for me? I think you should be. No, yours is HDMI.
transfer the presentation to another computer is possible, but mine doesn't have a battery. There's power here. So, I mean,
You guys are ready because they have a technical problem. Can you just, Belen, one of the few, please go so they can fix, they have a slide. The problem they have is copy their projects to another computer. Yeah, no Macintosh, no dependent
There's no HDMI here?
Should not, no? No. Doesn't
Look, just ask the guy in the box for an HDMI and we're done. All right.
So we have to. It's not working.
Não, baixo. Olha aí, bonitinho. Temos que arrastar ou assim?
Acho que eu preciso.
Are you connected?
Yeah.
Maybe I have to set something.
Não estás a criar
This put it in the audio and turn off. Yeah, but then it stops. It stops something, otherwise it gets stuck.
System.
What's going on?
Yeah, I don't know how to play very well with Mac.
Try to change the resolution. No, that's in preferences, maybe. There's the list of the
Normally it's just a
She tried to take these out. Okay. She tried to take these out, so it was
But I changed here. Isolation.
Lana Pipa. Okay, everyone, so we are going to show you our project, so some results we got and the idea of the whole thing. So we decided to call this project a robust encoding of self-localization in the hippocampus through the synergetic processing of eidetic and allotectic cues. So lately we have been having a lot of talks about the hippocampus, so, but just a quick tour among the whole field. So In 71, John O'Keefe has found that some cells in the dorsal hippocampus have some spatial selectivity to certain positions of the environment. So what he did was he first realized that by tracking the position of the animal along the arena and tracking also the firing rate of a specific cell in the dorsal hippocampus, it would be firing at a specific location of the arena. So what is called the firing field. So that was a nice evidence for what was the whole Tolman story about that the hippocampus would be the basis of a cognitive map that mammals would develop. So if we look at the structure of the hippocampus, it's based on a feedback loop connectivity, where the entorhinal cortex receives signals from the cortical areas. In there it projects directly to the dentate gyrus, so it's the first input area of the hippocampus proper, which lately projects to CA3 area. It again makes some filtering processing in here. It sends back to CA1, where CA1 then projects directly to subicular area and also back to earlier layers of entorhinal cortex. If we look at the type of cells that have been found within this region, since O'Keefe has shown the position specificity of these cells, we have now within the entorhinal cortex, we can distinguish it among the medial and the lateral side, where at the medial level we have what's called the grid cells. We've been looking at those for the past few days, where from a dorsal to a ventral level we have a different scaling within the activity of this type of cells, whereas in the lateral entorhinal cortex it's more based on a type of sensory information. So the animal would have some spatial selectivity based on the sensory inputs that it gets at a certain location with the environment. Those two project back then to the dentate gyrus, where sparse representation of space are already shown when plotted against space, where within the filtering of CA3 and the finally output region of CA1, we can see a more confined representation of the environment that the animal has been exploring. If we look at the spatial firing and how it is affected by changes, so sensory changes on the sensory or actually also physical changes on the environment, we see that at least at the very beginning of this morphing or this change in the environment, we can have some stability of the activity of the cell. So in the case A, a sensory, a cue, a landmark, a wall was removed from the north position to the east position. But still, the firing field of that same cell, it remained stable for at least for the first moments after this change. The same could happen, for instance, when there is an increase or a morphing of the environment. There is some amplitude, so some gain on the firing field as well, but still it remains coherent to the XY center of this position. And sensory completely changed on the sensory cues. We can see also some effectivity on the firing of these cells. So it means that even though it remains stable at the very beginning, there is a more remapping of this cell, or at least of this firing field according to the sensory change. So the same was shown by Goddard in '96 when they placed a rat on a box and it was running along a linear track. On that track, they were recording from hippocampal cells, and they could relate the place field activity of the cell regarding position of the environment. So just keep in mind that if a rat is in a box and just moving along this path, a path integrator would not be a high input within this system, so the rat is not moving, even though in the condition of light within the environment might have some optical flow input into that, and also some vestibular system input, but still the path integrator is not as accurate as when the animal is running. And what Goddard has found is that in the absence of light, the grid cell, the place cell activity tends to to a higher latency to understand or to correlate with the position where the rat is when compared to the condition of light. So sensory cues is also playing a role in the formation of place cells, but exactly what is the gain of each of the modalities has not been quantified, and actually it's what is lacking on the whole hippocampal literature is that so we have diverse opinions in how the place cell is formed, but still no one has quantified how one or to the other are interacting within the hippocampal structure and how it is affecting. So in our study, we are using a model of the hippocampus to study actually these two strategy modalities. So the sensory part or the path integrator, and how does it accurately represent the space in comparison to using either just one or to the other. And I mean, this looks trivial, but the whole point here, it would be to follow a quantitative measurement of these two inputs, which has been lacking in this field. So our methodology, it will be to emulate variable conditions for landmark navigation and path condition, and study the quality of spatial representation in the hippocampus. Mainly here we will be focused on the CA1 region. So as I mentioned before, there has been several attempts of getting place cells out of these two conditions. One has been by Hetois, where they use these objective functions, extracting the sensory inputs from a visual signal, and through a cortical hierarchy, they were able to get sensitiveness to a specific location within this environment. So, but that is using sensory cues. On the other hand, as we saw from the feedback loop of the hippocampal structure, also on the grid cell level, there has been several models. Lately, people have been focused on the path integrator approach, such as McNaughton and Guanella. And what they do is they take advantage of the path integrator of the animal in order to using attractor dynamics and bumping the activity accordingly to the movement of the animal, so that they can perform a grid activity. So in this case would be a square activity that with the bumping that would shift to a triangular tessellation pattern. So, but having these models, what allows to people is then to get place cells just from grid cells. Here is an example from Solstad, where a linear summation of dorsal to ventral grid cells would allow the animal to build a specific activity of a certain position. But still, we are not, so there is this dual these two types of studies of sensory or just spatial information, but still there is no understanding. So in our case, we planned to develop a model of the hippocampal structure that receives from both LEC inputs and MEC inputs, where through the modulation of the signals, we could quantify the spatial resolution of the place cells defined within here. A nice approach of this model is this arrow back from the CA1 region back to earlier layers of MEC, where we plan to use it as an error minimization of the grid cells. So we have been people have shown that deleting this back projection, we get rid of the grid pattern. So we plan to use this dual combination in order to then build a fixed or a strong map of grid cells. So Cesar can just show how.
So, the organization of our model is basically to integrate the input from the lateral that we assume to be the integration of perceptual information, basically landmarks from sensory cues, this kind of thing, and also the integration from the MEC that we use we use a grid cell model. So the main thing is, how do we actually integrate it in the hippocampus? And this is not exactly known. So we built on a model of the hippocampus. And actually, this is a model of the anti-gyros that could be put in a cascade up to the C1, but we are just playing here with a more general model. So somehow it's just using one layer. And it's based on this work from De Almeida with John Lisman, that they have shown the kind of competition that you have in the integration of the input in the hippocampus. It's basically, it generates this principle called E percentage winner take all, such that all the inputs are integrated into each of the cells. So each of the cells has a different weighted sum of the inputs that are coming. And all the cells are connected into an interneural network. This interneural network allows feedback inhibition that's more or less like that. So whenever one cell spikes, like in this case, it releases an PSP with a certain delay. This delay is very important because it allows other cells, like here, that they were also integrating their work close to the threshold to fire just after, while cells that were far away could not. This allows that not only the cell with the highest rate or the highest input fires, but also cells close to this value could also fire. With this methodology then applied to the rate, then we comes to what we call the percentage max, which basically at a certain point you get who's the most active input and you apply this transformation. So whoever has the highest input continues with the highest values, and you have a linear decay on the input until a point around 0.9 that it becomes silent. So the methodology to actually get from the grid cells in the original paper or in my work summing the LAC and the MAC to get the place cells is basically to sum all the inputs. This is the example of a map of sum of all the maps for one cell. And then you have this, compare this with the whole population and build a competitive values that you actually cut this landmark, this landscape, which actually gives you, this competitions gives you the place cells. So with this, if we can determine the activity from the MAC and also the activity comes from the LAC, we can make some integration and generate place cell activity. So the way we did, like in this project, we already had, this is a different paper that I showed that if you change the LAC according to the morphing of the environment, but you keep the MAC stable, you can explain an effect called rate remapping, that basically the place fields are stable, but the rate changes. What we wanted to see here is if we change what's going on in the LAC, or we change the precision of the grid cells, what would be the effect in the place cells? So for this, what we did is that we placed the agent like in a trajectory around the environment, and at 200 different points, we make it to stop, and it learns which are the inputs coming to it. And this creates a place cell in a specific cell. And we do this for 200 cells. So we can build a representation of the place cells with specific inputs from the entorhinal cortex, from the medial and the lateral. So it's basically what this equation is doing. So we kind of train which are these two values at 200 points, and we get 200 place cells. So our plan was basically that if we can, with this, generate this whole system, we can play with the inputs. Basically, we can modify some variables, for instance, the noise in the grid cell, so which is the integrated error. We can play with the representation of the lateral, which is basically the ambiguity and the specificity of the spatial map, which means how much information one single cell carries and how much information the population carries about the environment, about the landmarks. And also we can play which is the mixture between these two inputs in the hippocampus. And the metric that we are using is the position reconstruction error, which basically means how much do you can recover from your position when you're looking just to the output of the population in the CL1 or in the hippocampus. So the way we play with the with the lateral input, which is the landmark, our landmark representation, is that we can have different levels of unit spatial information. We basically is like, if you have a high, okay, this is inverted. This is when you have high spatial information, this means that one place cell, it's small, it's very specific. So whenever you have the activity of this cell, you have a high certainty of where you are. And whenever you have a very broad activity of the cell, then whenever you have this activity, you're not sure where you are. And also, you can have different levels of population spatial activity. So whenever you have low, which means that most of the cell fires at the same position, they are synchronized, which when you see all the cells, like consider each one of these is one cell, you don't know much where you are. You have some information, but not much. But if they are dissociated, so that each unit is representing a different position, then looking from the activity of all the cells, then you have more information. And this how this translates. This is rate maps from the lateral cells that we are generating. Having some example just regarding the unit spatial information. So for instance, here you have a cell that has high spatial information. So whenever, if it's very active, you have a high certainty that it's around this area. And these are two examples of cells that have low spatial information. So if you are just consider the activity of this cell, then you know that you're probably not here, but you're not really sure where you are. So you have to integrate all of them. And for the idiotech input, which is the proper perception, we based on the model of grid cells from Paul and Guanella, that basically has this kind of sheet of triangular tessellation that creates a bump of activity that is modulated by the speed. So it's basically, it has, every cell is connected to its neighbors, and by creating a modulation on the speed, you can make this bump activity just move around. And what we do here, we can put an error on this velocity signal. So basically, this is an example in a single dimension when it's doing a curve. Like when you have low noise and you have made the integration, you would always come up to the same place. But when you have high height, it integrates the error, so it finds that it's in a different place, and different trials goes to different places. So here's an example of the rate map of the cells with different levels of noise. So here, with no noise, that you can actually add these grid cells quite nicely. And here, when it starts to add, like 1 standard variation and 2 standard variations, when things get quite nasty. So here's an example of place cells that we can generate with this method. So we don't have much control. We made it completely automatic somehow and random. But you can see that some cells, they get really specific on the spatial information. So our method is basically to take in a condition without noise, all the maps, and use this as a reference. And from this, we can make some position reconstruction error using like things like David Resch said. So, which in this situation, like the methodology is, we are just worried about the maximum likelihood of the position. So we just take the distance of this activity to the rate map and take the position has the most value. So here, some of the results that we actually get. This is a not surprising one, but still shows that, at least shows that the implementation is fine. That this is a plot of the mean position reconstruction error and compared to the error on the speed vector. So we consider two conditions when we are only using the MAC input, which means the grid cells input, to drive the activity of the place cells. And then when you have a higher error, it goes up to the chance level of the chance level means that you are just comparing two positions completely random, and this is the mean error you would expect. And this is a condition when you actually use the feedback from the place cells that is also controlled by the LAC. So even though you still have a change in the error, it gets kind of controlled, so it doesn't go catastrophic to completely random. You still keep the information here, basically because you're using this feedback loop to anchor your representation in the landmarks. This is very preliminary in the sense like we still have to get much more samples to take this. But what this says that, okay, if you add the lac information, if you have landmark information, it helps your representation if compared to just using the path integration. And then this is then a representation of how the position reconstruction error goes when you are changing the input from the lac. Basically, how you are changing the landmark information into your system. Here we are considering kind of mostly just the lac input, so just to show which is the baseline. And here I can control ambiguity, which is basically what information you can get from the population level. And here is a resolution, which watches the size of the place field. So basically, when you go to resolution of 1, which means one cell it's not spatial active completely, then you completely cannot recall the position only from that. And what we see is that if you lower the information in the population, then the level resolution you need goes also lower. So what we wanted to do, but at the end we still have some work to do, is to show, we want to show that using the MAC input, you can actually play with this image and make it better, to show that one helps the other. So the future work, we actually want to quantify which are the effects of the two strategy against the others using this position reconstruction. We can easily show that MAC reduces, no, we want to actually show that MAC reduces when you're based on a lag. And this has never been quantified. And that lag to not lag, this is something that was already shown, but we want to have it quantified in a way that we can play, for example, we can cue the feedback from the hippocampus to the grid cells, and we can predict what would be the error or how would be the population. And this kind of experiment that people can easily do, but they didn't do yet. And just a matter of, for example, put a rat with this lesion MAC and in different environments with different cues and see how the population changes and this kind of thing. And also play how is this mixture going to be. And it's basically that.
To use the place cells only as some sort of readout of the LECA and MAC, the lateral medial entorhinal cortex, no?
It's a readout, but it's also a way to anchor the grid cells.
Okay, but you use it at best as a communication channel between the two systems.
Yeah.
Right? So, but in themselves, there's also an operation taking place at the level of the place cell. Look at CA3. CA3 will be sort of massaging these inputs as a low-pass filter, if you want, in some sense. But now you have not considered that effect, right?
Yeah, no, in this case,
right?
not yet.
So, but still, we have to look at this carefully because they're connected in a recurrent loop. But certainly the CA3 stage of that loop is doing something to these signals,
no, definitely.
which might give you more robustness, I would bet.
No, definitely, because then you can have like attractor dynamics
right,
inside.
Exactly. Now, and John was telling us that the whole model was wrong because grid cells actually don't do what we think they do. So is it going to affect your results?
No. I don't think so, actually, because the fact that you do not have grid cells doesn't mean that you don't have spatial type of cells already in the layer 2, right? So the problem here is whether those type of signals are accurate or robust along the track. So I don't think that would affect.
Okay.
No, yeah. What I think is like whenever you have a great idea and when you face the first small band, people think, oh, it's everything wrong. So like it's there. There are several models to explain many of the features, so they're not.
No, but look, but it might be interesting to also look at variations of your medial entorhinal cortex response that corresponds to these lesion and perturbation experiments and on these sort of non-grid but still spatially specific cells to show that it's actually identical.
Yeah, exactly.
That would be my bet.
Yeah, our idea is that it can be an interesting study to actually show that you still might need a grid cell system, but if you knock it out, there's some predict.
You can boundaries. Look at the boundaries. Like, what's the minimum amount of spatial information you need, irrespective of how it's topologically organized, right?
Yeah,
that's really cool. Very nice. So, Ivan.
In the graph where you're changing the leg and this one, are you sure you're only changing one one factor from one to the other? Because MEC only means that you're only using the grid cells, and MEC plus leg means that you're using grid cells and the other input and also the feedback.
Yes.
Or not, or because in the previous one there is no feedback.
Yeah, it means that you are using the feedback, you're using the lag and the feedback, and here, this is the true extreme cases. Like that's the point we want to play with these medial parts. Like, okay, you can have the lag also working, but you don't have the feedback. But this is like the best case with lag helping to anchor and also having the feedback, against the case that you don't have a feedback at all, because you only be driving by your own information.
Giovanni.
Do you have an idea what's the optimal mix of the two signals?
That's what we want to.
No, sure. No, exactly. But did you make like some sort of analysis to check what's the optimal, let's say, alpha for the complementary filter?
No, we just run like the best against the worst and see, okay, the best is better than the worst, but we didn't see in the middle.
I remember there is like some.
There is this
70-30.
Yeah, 32%.
Yeah, this was prediction based on the rate remapping data, that we could predict more or less that you would have 70% based on sensor information and 30% based on path integration, which if we find this here would be amazing. But,
All right, so look, thanks a lot, guys. Very good. Then you might have noticed it's getting later. It's 2:30 now. We have one more group to go. 2. Ooh. I thought 1. Okay, good. So I propose we just finish it and we have lunch, yeah? Okay? So let's be efficient, because we're here to learn stuff. So, where's our group? As I just said, we finish and then we have lunch. Read my lips, you get lunch. Okay, good. But first we have to do the presentations. All right. Huh? You can plug me out. Just kick. I need it. No. You need it. Okay. You have to. There you go.
Just this one, no?
During presentation, no. It looked like a
Yeah.
It looks like, okay, I'm not in control. Because drinking is like,
Yeah.
this is like when you're like to all right. Belen, Martina, Brigitte, and Irene.
All right. So our group wanted to explore also a field that has been studied also for some time. It's not something very, very new, but we were quite interested because we're coming from rehabilitation side. So for us, action selection is really important. And we wanted to see if we can influence action selection or selection patterns through reinforcing like one limb or another by means of scores,
and by doing that, like, yeah, influence what the subject is going to choose as an action selection. No, it is not. So, why we want to do this? What is our motivation to do this? When you have a stroke, there's some part of your brain is not working anymore as it was supposed to be, and there's some various conditions that you can suffer from. One condition that is quite common and that has not been studied so much so far is the learn not use. So what is learn not use? It means that when someone has an injury and cannot perform the actions that he used to perform before, he might choose two paths. He might try to use his affected arm and fail, and that's why he suppresses this behavior to use the arm, and that's why he learns not to use it anymore. Or he just compensates for the lack of his his affected limb and uses only the healthy arm that he has. And since he's using only his healthy arm, he gets a positive feedback from using his healthy arm. This is reinforced, so the less effective behavior is strengthened. So how can we undo learn not use? First of all, it is studied that it can be reversed. Depending on what the patient has, it can be reversed if you apply the proper rehabilitation. And one possibility is actually to reinforce using the affected limb in a positive way. So give him a positive feedback so he continues learning that he can use his arm. So what is crucial to make, to change his decision to use his arm, we have to alternate his past experience. We have to change the way, or change the expected reward he gets from selecting his impaired limb. So we have to alternate past experience, and we can alternate that by modulating the reward he receives. And we think that is more effective than do the other way around, which could also be possibly like force him to use his affected limb. So what we also saw is that a decision-making process is linked to, is a parallel process. So it's linked to perception, cognition, action, and thus goes in hand. And reward is an positive cognitive variable or cue that can trigger the decision-making process. And we found an interesting paper that actually studied the reward effect on motor evoked potentials. And we could see that reward could tune even the motor activations itself. So while the setup was quite similar that we are going to do in our experiment, the subjects were healthy ones, were trained or reinforced to use more their left hand. And what they saw that the reward, so they received higher reward for the left hand. So they then measured the motor evoked potential, and what they see that it was not like a linear progression of the motor evoked potential, but it was alternating, that it already started on a higher level. So using reward could already alternate the decision process in a very early stage. So therefore, we think, or what we want also to see in our experiment is by taking or looking at the muscle activation itself by using EMG, is reinforcement a successful strategy? What have we done so far? We've been working on a rehabilitation gaming system, which is called RGS in short. RGS is a virtuality-based tool for stroke rehabilitation of upper limb motor deficits, and has been applied already in various clinical studies, where we trained with patients who perform certain actions and use their both limbs, or alternating the both limbs, and rehabilitate that. How does RGS work? Well, the paradigm that underlies this is the action, observation, action, execution. So you observe your own action that you do, and you actually also do the action. And it is shown that this method does optimally drive functional recovery because it uses the paradigm that our brain remains plastic all over his life. So it can learn all over his life and rearrange itself. And it does so by using the mirror neural system. So the questions we had, like from the studies we've done so far and what we've seen or what we want to do, like reimbursing the not use. So can we, by positive reinforcement, which we're gonna do by alternating or augmenting the scores, can we affect action selection patterns so that you use one limb more than the other, so that the probability of selecting one limb, for instance, would go up over time? And the second one, which level do we have to apply? Because what we know, okay, we can maybe influence this reinforcement, but how much do we have to, how high this reward has to be? We don't know. This is not known. And we want also to see what influence has this reinforcement later on the muscle learning. That's why we want to try the EMG, if we can get some data out of that, which would help us in answering these questions. And the other part that is very important, especially in the rehabilitation terms, is when does the decay of the learning start? So is there any decay, and when does it start? Because it's very important to know how long a rehabilitation has to take place, how long the patient has to be exposed to a reinforcement to make predictions what the prospective outcome is going to be. How we wanted to test or how we wanted to answer our questions. We decided to design the following experiment. We had 3 blocks with different scores where, well, the scores were the same but different levels of reinforcement of the scores. Like they were cascading. Like there was once we did reinforce it twice, 3 times, or 4 times. And we had, before these 3 blocks started, we had a training session, so the subjects would get used to the system itself, and a washout where we brought them back to normal again. And we were discussing how to, should we only reinforce one limb, or should we reinforce only the not widely used one? So, but we decided to reinforce both limbs in an alternating way. So every 10 trials, we were shifting the reinforcement to the other limb. So we would reinforce both limbs in one block. Yeah.
Okay, so just something we found curious and we wanted to explain you is this working? Okay, so you see the rule we defined, it looks very simple, right? Like we first reinforce one limb for 10 trials, then we shift to the other limb for 10 trials, then 10 trials. And in a first place, we thought, okay, this is going to be too simple. So we humans are so clever in action selection, and they're going to find out, right? So we decided to go for probabilistic rewards. 80% of these trials would be reinforced, and then 80% of these trials. It was a complete mess. The first subject we tested, we realized that it was not working. So we are not that clever. So we went for 100%, no probabilistic reward. So we were keeping the rule very, very simple. Regarding the setup, as you see, we were using Armio to capture the movement of the users, specifically joint angles for elbow and shoulder. And this task was very simple. So there was, well, we instructed participants to locate their hands over a home button, And after that, a target appeared always at the same place, okay? And they had to choose freely one limb or the other to reach this target. Then after the trial, we were showing scores, and these scores were computed as a function of how fast they were and how accurate they were, multiplied by the amplification factor, if any, okay? So as you can imagine, there was some variability because it was depending not just on the amplification, but also on your performance, the reward or score.
So in the rewarded trials, there was amplification, and in the non-rewarded, there was no amplification?
No amplification. The score was just function of speed plus accuracy, but not multiplied by any factor.
The amplification was as well?
The amplification was, depending on the block, we tried 3 intensities, let's say, of amplification. So 2, 3, or 4, one of these possibilities. And these blocks were randomized, the order of these blocks.
So yeah? Would it be very clear whether it's amplified or not?
It should be. When we were trying it, it was very clear. That's why we went for the probabilistic reward. But then when we tried with the first subject, we realized that it wasn't clear. So, well, and we will see from the results that it's even more interesting. They didn't find out this rule. Okay, so basically we collected data from different, yeah, it was funny. Yes, he was a bit scared. You can see his face that he's like looking, oh, I'm afraid. But okay, well, we collected data from different sources. First, as I said before, Armio, so joint angles. Second, we used EMG. So we wanted to capture muscle activity in deltoid and biceps, just for one limb. Also, we used data from the scenario itself to capture some data about performance, so scores, which hand they selected for each trial. We used the Edinburgh handedness inventory just to control for the dominant hand of the participants. We wanted all of them to be right dominant. And at the end of the experiment, they completed an online awareness questionnaire. We wanted to check if they were aware or if they discovered the rule, the hidden rule, or if they found any pattern in the reward rule. So we tested 7 participants total, just 3 of them with EMG. They were around 30 years old and right-handed. So let's go for the results. This is the interesting part. So, okay, these are the questions we had in the questionnaire. And the nice one was this one. So we asked, is there any hidden rule that you found across the 300 trials? And as you see, the rule, when they explain it to you, is very simple. They said, yeah, there might be some hidden rule because you told me in the beginning. No? Because we said, when we gave the instruction, we said, there is some rule, some pattern you have to discover, so you will select one action or the other, and your goal is to optimize your scores, to get as many as you can. But none of them discovered the rule. They just invented their own rules, and each of the participants told us a different story, very different. So they were unaware of the rule. It looks they were unaware. But they were consciously unaware of the rule. Because when we look at performance, here we are plotting a percentage of correct actions, okay? We define a correct action as selecting the limb that was reinforced in this trial. This is a correct action, okay? Doesn't matter which limb. So we see that when the amplification was 2, here we are centering the trials in the shift. So 0 here in trials corresponds to the first trial in which we shift the action outcome contingency, okay? So we are shifting the reinforcement to the other limb. And we see that the probability of performing a correct action is around about 50%, but then it drops after shifting the reinforcement, okay? So it means that somehow they were following the rule, but they were not aware of the rule. And this effect is greater for the greater amplification, as you see. The drop is even more accentuated. So next, we looked at reinforcement history, and I explained you this. Basically, we did a logistic regression, taking as a predictor the score, okay? So from the score, we try to predict which is the probability, or if you are gonna select one action or another one in the next trial, okay? And then we get alpha. That alpha would be the coefficient of this predictor, okay? And would give us an idea of the influence of the reward on the next action, okay? And we do this for a trial lag of from 1 to 8, which means the reward I just got in the previous trial influenced this amount my action. The reward I got 2 trials ago influences with this intensity my current action now. Okay, and you see when we give an amplification of 2, we don't see that there is much retention of the influence of the previous reward in the previous trial. And neither here, we can see here a bit of drop, but here we have a very clear pattern of the retention of the influence of the reward. And we can see that more or less it takes 3 trials to disappear. So it may be that the intensity of the amplification is correlated with how long we retain the effect of this reward in the next trials. And finally, we looked at reaction times. We didn't find anything regarding trials before and after the shifting the reinforcement. As you see, it's just a line. But if we compare the 3 different amplification conditions, we see that reaction times were lower during amplification 3 and 4 when compared to amplification 2. And this may mean that when we have a greater reinforcement, we have a faster reaction time. We respond faster. So now Puneet will present results from EMG data.
So in 20 trials of the starting of a training period, we recorded the EMG 20 trials, and in the session of a washout, we recorded 20 trials of EMG. This is a 2 graph. Blue graph shows that the activation of a deltoid, interior deltoid, and the red color shows the biceps. And this is a 1 trial. This block is a 1 trial. So we recorded from a 20 trials. So this is a very crude, all of the picture is clear here in the muscles memory. So you can say that the red color is the graph of the activation when there was in the training period. This is a graph of a bicep, the activation pattern of a bicep when there was a training period, initial 20 slides. And this was for the deltoid. The blue color graph is a deltoid. So they are activating more muscles when they are training. And this is a graph of a down graph, is like after the whole session is completed in the washout session. So washout session, you see, they are not firing. So somehow the brain is optimizing the muscles memory. So they are optimizing the firing rates of the muscle. And you can say that from this plot, so after the reaction time you can see. So these are firing previous in the slide. So this is the reaction time. It shows the indication of a reaction time. When they are trained well, so their reaction time is increasing. So in this context, the muscles memory plays a lot of a role in the rehabilitation. So this is the conclusion.
Okay. So, basically this was just preliminary data, and we wanted to use EMG to see the effect of the different rewards we used, the different amplifications on muscle activity. But we couldn't reach that, but this would be very nice to follow now, and we plan to do it. Basically, as a conclusion, what we see is that we can claim that higher rewards or higher amplification lead to a shift in action selection and lower reaction time, as we saw in the graphs. That muscle activity is optimizing, as we lately saw when we see that the activity is reduced after training. And we see that the effect of reward reinforcement is retained longer when we have an amplification that is greater. So it may be really important to find out how much amplification or how much amplification in reinforced therapies we need in order to retain longer the effect in patients. Because maybe what we could do and could be very interesting is we amplify, we reinforce the use of an action, and then just after that, since we know it's gonna be retained for some trials, we just augment the error, which will counter-effect the amplification of the reward, but they will learn very fast from the error because it's amplified. So we can play with these different parameters. And what we learned also from this experiment is that even though we were looking for retention, and this is what we want for rehabilitation, we seem to be very adaptive. Just in 3 trials, we lose the effect of the rewards. But, well, this is how it works and probably is better for our survival. So that's the story. Thank you very much.
Great work. Questions? Okay. Richard.
I wanted to ask about something that's not very related to to the retention time of the.
Okay.
So I find it very cool the stories that people would make to come up with the hidden rule, and you always observe something like that. Even like if you do any experiment when you have choice, everybody will always come up with some confabulation. So maybe there is a way also of exploiting like the extent to which this confabulation matches the performance, and that could give you because also the non-use of the paretic limb, right? It's also some sort of confabulation if it's a learned non-use. You tell yourself a story that it would actually be worse in case that it isn't. So maybe this confabulation that in this case doesn't allow you to find the rule could also be something that would make you use your paretic limbs more.
Yeah, it was actually really interesting because there were people that saying their strategy was using only one limb, only the right one, but it's not true. I mean, we saw in the log that they didn't do that. I mean, they told us that my strategy was to selecting one hand or another. I just use one side. And then you analyze the data and you see it's not true. They're shifting. And so they do it unconsciously. Right. And like for us, it's interesting to see, well, was it really because of we gave the reward and they were really not aware of that? So it must be a process that happens before, like before the awareness kicks in. And
with what is cool here in the confabulation part, there must be a systematicity to it. It's not random.
No.
It would be really interesting to try to investigate just that part of your manipulation in a more systematic way. Right? To, for instance, look at effort. How does effort modulate the confabulation? How does error modulate? How does some distractor modulate? It would be very interesting to see. Because confabulation, for instance, is really high-level cognitive process, like, okay, I think I'm Jesus, I can fly, and so on. But now you see that actually already in these very relatively simple tasks, people are trying to interpret, right, what they're doing, and it relates with also to Richard's task, right? Even though that's more the psychophysical thing, and we don't ask people about any explanation, but it would be an interesting aspect to include here. And the other thing that relates to this, it might be you didn't ask people about confidence, right? No. But you remember this anti-correlation between ability and confidence. It would be really cool to include this as an index to see if we can recover that there as well. Right? It's a beautiful paper, especially relevant for students. Like a student also means you're ignorant about many things, right? Otherwise you're not a student. And the paper is called Unskilled and Unaware of It. And that means that it shows an anti-correlation between skill and confidence. So those that have no skill are very confident. This is what it shows. It has to be replicated many times. And it holds for many students. So overconfidence can prevent you also from learning stuff. So be careful with that. Anyway, it would be cool to include. Great experiment. Other questions? All right, thank you very much. Wonderful. Oh, just a bit on motor memory, the muscle memory I didn't get, but you can explain to me later. Yeah? Great job. All right, the last group is up. And then
we run to lunch.
No, wait, then what we will do, I will quickly finish up with the outcome of our song festival. I'll be quick on that. And then we'll do the award ceremony during lunch. Is that okay? All right, cool. Grab the prize.
Go.
No, for the song festival.
No,
come just 1 second, because here we have the presentation and it isn't shown there.
Ah, so,
Do you want to try? Come on, Alberto.
Is it your computer?
No.
No.
That's why. So where is
What is
screen?
Is your field?
Where is screen?
Don't know. I mean,
Display.
Okay.
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 Maybe interface screen. Okay.
So, hello everyone. Our project is about spatial navigation and spatial memory. And the spatial memory is a process that enables to encode, store, and retrieve information on spatial location and configuration. The aim of this project is to assess if two different modalities of navigation, so the active modality and the passive modality, can influence the recall and the learning, the spatial learning in an unfamiliar environment. It seems obvious that a subject that can explore actively a new environment can improve his spatial learning compared to a subject that is only passive exposed to it. And there are several factors involved in active spatial learning, as motor and vestibular information, the possibility to take a decision about the directions and the path, obviously the attention, and the capacity to manipulate acquired information. There are several studies on this topic that tried to assess the influence of these two modalities, but the results are inconsistent because for the majority they didn't take into account the presence of a neutral environment or the locomotion. And here comes the idea to use this beautiful space that is XIM, and is apparatus for the mixed reality to perform this experiment of spatial navigation. This is a room of 25 square meters with a sensitive floor that is sensitive for pressure, and a system of tracking, and 4 walls on which 8 projectors can project any kind of 3D environment. So with XIM we had a semi-natural environment for this experiment.
Okay. Well, this is an image of the XIM in another experiment. But in fact, the experiment we are performing, it's a follow-up of an experiment that was performed here in SPECS in 2013. But we have implemented some things that we will tell you later. Okay, I will start with the description of this environment. The subject will be situated in a house which is in a neutral landscape with green field, blue sky. Then the house has 4 rooms, as we see here. The subject enters the first room A, and depending on the mode, it can navigate freely or it's automatically sent to the other ones. Then we have each room has a different wallpaper, and we call this a global feature. And then in each house, there are 10 objects, which are distributed. Okay, sorry. Okay, there are some screenshots of the house. And okay, our experiment. We perform it with 8 subjects. We expected to do it with a bit more, but because of time, we just have this for the moment.
which were equally divided into groups. Some did the passive mode and some did the active mode. Then our hypothesis was that the navigation mode would influence the memory, and we would test this with a test at the end of the experiment. Then we see that the independent variables are this active or passive navigation. And then we have some dependent variables, as we see here, this spatial memory recall, some behavioral data that we will tell you, and, well, okay. Then, so the subject comes and it doesn't know if it's in the active or in the passive mode. And these navigation modes consist on, okay, in the passive, they are free to move in the room, but they are automatically sent to other rooms with fixed time for each room. And on the active, they enter the house, but they can step into the room they want by pressing the tiles on the floor. But the total time spent on the house is the same for both modes. Then, we gave the subjects some consent form and instructions before the experiment. Then, before performing it, we had to calibrate the gaze tracker, which this, and then the subject was sent to the exam, and it started the experiment. And depending on the mode, would happen one thing or the other. And when finished, there was a digital questionnaire, which Martin will explain you, that served for looking, well, if they had a good memory or not.
Yes. What we have changed in the experiment now in comparison to the experiment in 2012, we have 10 possible combinations of randomized local and global features. Local features are the 12 objects here. And for global features, they are here. These are the wallpapers. Then in the other experiment, you can look between the rooms. So we have closed the door. So you see only one room with the objects in this room. Then we added the IMU to tracking the head gaze, and we have implemented a digital questionnaire. Yes. So after the experiment, the subjects are presented the top view of the house, and then they have the objects here and can drag and drop and places the objects in the top view of the house. Then we have the wallpapers. They have the subjects have to place the wallpapers in the middle of the room. That means that this room is colored in with this wallpaper. After that, the program saved the positioner of the objects and the wallpapers, and calculate which room and which position in the room is chosen. So we have as the result of the questionnaire the scene in the experiment. We have 10 different scenes and the position of the objects. the calculated room which is placed, and the slot of the object. This is better for the evaluation after the experiment and the result presented. Valentina.
So some preliminary results. We found that global features were not different between the passive and the active subject, and we calculate this assigning a score for each subject, for each feature, global or local, set in the properly or semi-properly position. So these are the means, and we found instead a significant difference between the means of the score in the local feature between passive and active. In fact, the passive performed better than the active subject, and also we found the same significant difference between passive and active, considering an overall score, so considering local and global feature together. And so we confirmed our first hypothesis for at least for the local feature. We didn't find a difference between the global features, and of course there are some improvements to do to the experiment. For example, the number of the subject that is still too low. We had 8 subjects, and we have to analyze the data collected from the gate's position and from the tracking of the trajectory. So these are the maybe it will be important to consider if emotions can be influential for the spatial memory too, or consider the time in the test phase to recall some object, global or local, and so consider also the attention, and run more subjects, of course. And this is Alberto's backstage this work. Okay, thank you.